Alright. Recall that the reading guide is due tomorrow, 39. And the lab report for Piccolo is due Friday. Your test on 20, the 2 questions on 21, and then
It's already
I know. 36 and 39, the test is on Monday. We got to go fast because you don't have school next Friday, and then we got to do 30. I want to do at least in at least one revision chapter before y'all go for spring break. Yeah. We did this, yeah?
Yes.
We did this?
Yes.
Here?
Yeah, that's pretty good.
The more surface area, the more plants will be. So the sign, that's all that we care about that. Most roots, so plants that roots go in the soil. The soil provides water and minerals for the plants. A lot of times most plants have a symbiotic mutualistic relationship with fungi. These relationships are called mycorrhizae. And what it is that the fungus, you think of a fungus, you think of a mushroom, right? That's the reproductive structure of a fungus. The majority of the fungus body, it looks like this. It's called something called mycelium, and it's underground. Or if you ever turn down in the forest or like somewhere where there's leaves, and turn over leaves and see all these white things, you're like, what is that? Yeah. That's a fungus. It's the mycelium of a fungus. Most of the fungus is underground. Anyway, fungi are cool. They secrete enzymes into their environment to digest stuff, and then they absorb it. That's kind of how they are. But in this case, the fungus, what it does is it creates a wrapping or a sheath around a root, or it can actually stick its little mycelia inside the root to get sugars. Hold on. Like this. So here's like a sheath around the root. And the mycelium in this case would be the purple. And it sticks a little bit of the inside of the root itself, but most of it wraps around it. Some mycelium actually just go directly into the root. What it does is that the mycelium or the fungus benefits because the plant gives us sugars and organic compounds. Why the plant benefits is because the mycelium and the fungus itself increases the effective reach of a plant to a much higher surface area in the soil. It provides the plant with more water and more minerals. It makes the plant healthier. Most plants have mycorrhizal relationships in the soil where they grow. Most plants do, 90-something% do. And they're much healthier with them. You can grow a plant with and without a fungal mycorrhizal association, and the ones grown without it are much shorter and not as healthy. So anyway, that's an example of a symbiotic mutualistic relationship. Yeah. This is chapter 36. I sent this last week. I sent you 39 today, which we won't start until tomorrow. Maybe I sent both of them. I don't recall. All right, so transport. Let's review transport in plants, but also helps us review transport in general. So remember, there's active transport, which requires energy, and it moves solutes against their concentration gradient, or up, right? And then there's passive transport, or solutes that can diffuse down their gradient directly through the cell membrane. It's full passive transport. These things are what? In order to move just to the membrane, they have to, what's their properties? They have to be non-polar, right? So they have to be hydrophobic. So they can pass through the tails in here that are super hydrophobic. Solutes that are hydrophilic, they're ionic, or they're polar, moving down their concentration gradient can't pass directly through the membrane. They have to move through some sort of protein carrier. So we call this facilitated diffusion, where they pass through a protein carrier. Remember all this? No energy is required for any of this. So, why do we care about this? Plants are constantly transporting things. So they have to take in minerals and water from the soil, and they have to transport it off the plant body. And then plants make organic compounds, so it has to be transported to the rest of the plant body. Right? Remember, selectively, all cell membranes are selectively permeable, which allows certain things to move in and certain things not to. So if something is hydrophilic outside of the plant cell, and there are no protein carriers, will it move in? It won't come in. That's what makes them selectively permeable. Okay. So, you need to know about a proton pump. Plants use proton pumps quite often to establish an electrochemical gradient across their cell membrane. Most animals use sodium potassium pumps to do the same thing. The plants use proton pumps often. They pump hydrogen ions out of their cells into their environment, generating its active transport, generating a gradient where it's more positive on the outside and more negative on the inside. It's called the electrochemical gradient, right? These are called proton pumps. And that's called the membrane potential that it generates. Y'all remember this stuff? Yeah. Okay. It's actually good we review this because we're reviewing chapter like 6 or something a long time ago.
Hey,
We learned a lot. I know. I'm gonna miss y'all. Although I feel like we'll never get to the end. I'm tired right now. I'm cranky too. I'm so hungry. I really want to eat. Yeah. Oh, I actually can't eat right now. I'm having Crohn's problems. And like, it'll make it worse if I eat. So I just thank you, though. So the proton gradient is used by plants for a number of things. One is to, oops, I don't have another slide on it. One is for the picture at the top, to help take up cations from the environment. Cations are positively charged ions other than hydrogen ions. So by pumping out the hydrogen ions, it creates this gradient and then allows things that are cations or positively charged to move down their electrical gradient and can move into the cells. It helps roots take up extra ions within through the soil solution, what we call the watery stuff in the soil solution. Can also help plants take in ions that are negatively charged and maybe scarce in the environment, but not use energy to do that. So this is an example of something called co-transport. So it uses the diffusion of hydrogen ions down its gradient through this protein called a co-transporter to couple it with the movement of a nitrate ion up its concentration gradient. So this is how plants can get. Now plants need nitrate to help make it proteins and nucleic acids, right? The nitrogen. So. Yeah. Mm-hmm. And then they can also use co-transport to take in things that aren't charged, like monosaccharides, like little sugars and things, going up their concentration gradient as well. This is how plants take up sucrose. I think this is sucrose. What's the disaccharide? Okay, now we have to remember osmosis. So these are the movement of ions. How does water move? Through osmosis, right? It's passive transport. It moves through a protein channel, however, called an aquaporin, right? So plants and their roots are constantly taking up ions out of the soil, which is the direction that water will move. Into the plant. That's how plants actually increase their taking in of water, by moving in solutes, and then water just follows via osmosis. It's also why you can't water a plant with salt water or with a water that has a lot of solutes, because there's a lot of solutes in the water that you're watering it with. What direction will water move? It will leave the plant and you'll dehydrate it. So osmosis is the diffusion of water down its gradient, or down its own gradient.
I remember.
Remember the YouTube. We're not really going to use these so much right now. What we are going to talk about, however, is water potential. We will review this. We talked about this last semester, but this is super important because water potential we talk about with the plants. Remember, water potential is a measurement that combines what we call solute potential and pressure potential. Are you okay? My son does that in the car, so I'm like, talk to me, say something. What is that, mommy? Okay. Because if you can't move air, that means you're really choking, and then you have to be like,
That would be like what?
do the Heimlich thing. The Heimlich thing. My friend Amber had to do it on her husband on their anniversary. She was like really choking, and she had to like Heimlich him. Be like,
All right, so when somebody's choking, I'm gonna be like, there.
Okay. So solute potential plus pressure potential. You need to remember that water flows from an area of high water potential to an area of low water potential. So let's review a little bit more. Anytime you add solutes to a solution, the solute potential goes negative, goes below zero. So let's go over here. Here is a glass of distilled water. Distilled water has no solutes, and this one is open to the environment. So we say the pressure potential is zero because it's open to the environment, and the solute potential, oh, shoot. Sorry, hold on. Okay. No solutes. So pressure, water, we say distilled water has a water potential of zero. Moving over to a plant cell that is flaccid, this guy's like not super happy. There are solutes in all cells. There's nucleic acids, there's carbohydrates, there's proteins, there's solutes. So the solute potential will be a negative number, -2, let's say. Pressure potential in this case is zero because the cell is flaccid. You take this cell and put it inside the distilled water. What direction will water move? Area of high water potential to an area of low water potential. So we'll move into the cell. Until what happens? In a plant cell, now in an animal cell, it would just pop eventually, right? The plant cells won't because of their cell wall, right? And their cell wall is really rigid. And at some point, the pressure that, remember the water will be taken up by like the vacuole, and the vacuole will get big and big and big and push against the, here. So, here. Okay, so the vacuole prints against the cell membrane, which will press against the cell wall. And at some point where that pressure basically negates each other, here, the pressure potential is positive and the solute potential is negative, then the water potential of the cell will be zero. And if it's zero, then we say the net movement of water outside the cell to inside the cell or vice versa is zero. So water will still move, but the net movement is zero. Make sense?
Oh, so it's like equal coming in.
Yeah. So we talked about this. Okay, I got that there. So solute potential is this one. Anytime you add solutes, it goes to negative. The more solutes, the more negative that number will be. It's also called the osmotic potential. So pressure potential has to do with the cell wall if we're talking about plant cells. And it's the Turgor pressure and potential. Or in Turgor pressure in, I'm trying to say. For example. Anyway, it's the pressure that the membrane exerts against the cell wall. Yes.
You said that
Solute potential? It's the number of solutes that are in the solution. So it could be in or out. That's to come. So if there are solutes in this water, this would have a solute potential. The cell? What do you mean? Solutes aren't moving here. We're just moving water.
okay.
All cells, what I'm saying is all cells have solutes. Like because they have dissolved. That's what's already there. Okay. Are you good with this stuff? We talked about this all. Okay. tubes. We don't, like, this is not a plant cell, but blah, 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 blah. If you put a plant cell, so here's our little plant cell, open to the air, pressure potential 0 is flaccid, solute potential is negative 0.7, stick it into a sucrose solution. It has a solute potential of negative 9. What direction will water move? From high to low water potential. It'll move out of the cell and leave. So this cell will not be turgid. It will become what we call flaccid. And eventually it could plasmolyze. Plasmolysis is when the cell membrane pulls it away from the cell wall. That will kill the plant cell. You remember this stuff?
Yeah.
Okay. Coming back. Good. Now, the opposite is true when you put it in distilled water that has 0 solute. It'll become very turgid. And that's what is to help you. Right? Any questions on this stuff? I'll kind of review these. Okay. So when plants become flaccid, they will wilt. Like what you actually see is a wilting plant. When you see a plant that wilts, like water it immediately. Eventually it will die. Why do plants need water? For photosynthesis, the water is split to donate the electrons. Yes, it gives it a light reaction. If it runs out of water, the plant's trying to photosynthesize, it can't do it, it will die. Because they don't eat, they make all their organic compounds. So, remember aquaporins, the channels through which water moves. It's still passive transport. Yeah. Okay. So, transport in plant cells is also regulated by the structure and the compartmental nature of plant cells. So, plant cells, when you line them up end to end to end, they kind of look like this. They're like little boxes. See that? And there are, so let me actually, hold on, I'm going to come back. I'll come back to this. But here is a root hair. See the root hair? This is where the absorption takes place in a root, on the root hair. So we talked about root hairs last week. Have we mentioned root hairs? Okay. I'm going to go back. Another PowerPoint I'm going to send you is chapter 35. We did talk about it. Did we talk about nematophores and like tubers and... No. I don't recall. Did we talk about him or no? No. Oh no.
Oh, yeah.
We didn't talk about this stuff at all? Oh,
No.
God bless. Oh no. Okay. Well, I'm gonna come back to this in a minute. Let's find our root. Hold on. Okay, so root hairs. I'm gonna come back to this PowerPoint. I will be sending you this PowerPoint as well. You have to understand some plant anatomy to order to really understand what's going on in chapter 36. So we used to have a giant plant unit 35 all the way through 39, all inclusive, but they deleted a lot of the info that you need to know for the AP exam. So I deleted it, but we still kind of have to know some of it. So in a root system, the entire root system is not for absorption. Only the very tips of roots where we have root hairs is where absorption is taking place. As the root continues to grow and move through the soil, the root hairs will be the very end, the newest part. The older part of the root becomes sort of woody and hardened, and its only really function is for transport in like the middle of it. The vessels are still there, like xylem and phloem vessels, but for support, to hold the plant or anchor it into the soil. Okay? So root hairs are found, like I said, on the youngest part of a root, and these are little root hairs. And their function is to increase surface area, to increase the amount of absorptive capabilities that the plant has. That is where water and mineral absorption takes place. It's on the root hairs, not older parts of the root. So any of these types of roots that you see, these are all kind of crazy roots. We talked about these. But these roots really for structural support, these are really cool looking random ones. But you know, you're ever walking on a sidewalk and there's like a root that's coming out of the, you're like, oh, yeah, don't trip on it. That root right there is not for absorption anymore, it's for support. You see a tree fall over in a storm, it's like giant root ball comes up. So most of those roots that came up were really only for support now, to help keep that plant in the soil. Just the very youngest part of a root.
What's the one at the very top, like, I don't understand, like, what?
Some trees have roots that like fall off of their branches. It's kind of crazy. They're called aerial roots.
No,
They're
no,
really,
like that one, that one.
oh, prop roots. They help hold it up for support on top of the soil.
So if the tree does fall, is it gonna die?
Well, probably unless you're gonna be Superman and put it back in the soil.
Oh.
Imagine one of those giant trees.
Okay, so like the roots aren't in the ground.
No, they're definitely out.
Oh, okay, I thought like.
You would die.
Alright.
Let me skip. We'll talk about this later. Let me show you something. Take a side step. We have different types of tissue and us plants have different types of tissue in them. Okay? Their epidermal or dermal tissue is the blue stuff on the outside. It's basically like their skin, just like us, okay? They also have vascular tissue, sort of like our blood vessels. Their vascular tissue are where the water and minerals flow in and the sugar solution flows in. It's called xylem and phloem. In this picture, it's the purple, okay? And then everything in between is called the brown tissue. You don't really need to know that. The vascular tissue are the vessels, and they're called vessels, but the water, they're called xylem vessels, or the sugar stuff flowing, the stuff moves inside of. I want to get to a root stuff. Hold on. Oh, where's my root? What are you doing?
It sounds like
Annoying me.
it sounds like when you're at the dentist and they're sucking like the water out of your
It does. This picture is actually really phallic. I'm sorry.
that's fine. Oh, chill out.
You don't hear me over there? Oh. Whoever's recording heard that. Or watching. I should move that. That picture was terrible. Never mind.
What was it?
Nothing.
Can we see it?
How'd you not?
It's very, I didn't realize it was so common. This is the very tip of a root. See the bottom of the root? Okay. So a root, the cells at the very end are highly mitotic. Remember when we did mitosis, so you looked at on your root tip side because you saw the cells in mitosis, the stages. So as these cells divide, this root tip continues to move through the soil. The cells that are produced will mature and differentiate into the different types of tissues, the dermal tissue, the ground tissue, and the vascular tissue. On a root, some of the cells on the dermal tissue will grow extensions, and these are the root hairs. But as you keep growing up on the root, as the root continues to grow, eventually the root hairs will be degraded and this will become mature, and like I said, won't function as a like absorptive capability anymore. Anyways, I just wanted to show them. So here's our onion root tip over there where you saw the. Yeah. Okay. Back we go. Oh, I forgot. So as minerals and things, water, are absorbed into a root hair, once they come in, they actually can take different pathways. Okay, there are two pathways they can actually take. This red pathway or this blue pathway. The red pathway is called the apoplastic route. And notice it travels in where? The cell wall. Now, is the cell wall a selective membrane? A semi-permeable membrane, is it? Anything can travel in a cell wall. It is not selective. So the apoplastic route is pretty quick. However, eventually, before these chemicals can get to, or the solutes can get to the xylem vessels themselves, the plant needs to have a way to like screen them. Be like, you can't come in here, you don't belong. And we'll talk about that in a minute. Now, sometimes the minerals can go ahead and move into a cell membrane and be selected for right away. And that's if you move on what we call the symplastic route, this blue pathway. So it moves into the cell wall, and then it moves into the cell membrane and gets inside of the cytosol. And once here, it can travel from cell to cell to cell in the cytosolic pathway through plasmodesmata or the opening. See that? It just travels right into the vascular tissue.
How does the cell wall can faster?
Was not selected. Usually, like, imagine coming in, like, through the doors in the morning when there are a lot of people coming in, you have to wait in a line.
Yeah.
Versus if we had no doors and you just, like, could walk up.
I mean, but does it slow it down at all when it gets selected for and that?
It will be slowed down here, yes.
Okay.
So it's faster getting to here, but it will still eventually have to, like, be selected for. And it's random what solutes can travel and which pathway, honestly. It's random. So how does the cell actually screen these chemicals or solutes traveling on the apoplastic route? Because in the middle of a root, there is a structure called the vascular cylinder, which is what this is called. This is a cross section of a root, a vascular cylinder. And these big white balls in here basically are the xylem vessels, where the water minerals are eventually going to get to. All right. But this big, like, orange or red line around it is something called the endodermis. The endodermis is a ring of cells that screens all the solutes that are traveling on this apoplastic route. The endodermis, in this picture, is these orange red cells. How does it force them to be screened? There is a waxy strip surrounding all the endodermal cells called the Casparian strip. And wax is hydrophobic, right? So anything that's hydrophilic that's trying to travel on the apoplastic route gets stopped there. It won't pass anymore past this hydrophobic wax. So it is forced to travel into the cytosol of these endodermal cells if there's a protein channel. If not, it won't be allowed to move in. Could be damaged, yeah. And then at that point, it can travel into the vascular tissue of the plant. Okay. Yay, all that good here. Great. Those are the different routes. Your book talks about a transmembrane route, which honestly is ridiculous. That's sort of like if you're traveling to Charlotte, you would get off every exit just to get right back on the interstate. Because things traveling on the transmembrane route have to pass through a protein channel, into the cell wall, back and through a protein channel, into the next cell, back to the cell wall, back through another. It's like getting off the interstate every exit, which would take forever. So most solutes don't travel along the transmembrane. If they do travel on the symplastic route, recall that there's a vacuole inside all the plant cells. So what allows for solutes to move quickly around the vacuole? Remember what it's called? Close, cytoplasmic streaming. Remember, it creates its own little current inside the plant cell. So that's how they can travel quickly around the vacuole without having to diffuse into and then out of the vacuole. Do you recall that? Maybe not. Now you do. Great. Okay. So we say that the travel or transport of long distance transport of fluid, which would be the water and the minerals, or the sugar, which would be the phloem sap, we say it accomplishes something called bulk flow. Sort of like a pipe, stuff is moving in large quantities. So water and solutes move through what we call tracheas and vessel elements of the xylem. And they can also move through this, what we call sieve tube elements of phloem. Now, we have not talked about these yet, have we? So I'm going to skip back to my other chapter, PowerPoint, that I will send to you today. So let's talk about these. Okay, so these xylem vessels are what allow water and like minerals like nitrates and different ions from the soil to travel up through the plant. There are two types of xylem vessels, what we call tracheas, and the other ones are called vessel elements. All of these cells are what we call dead at functional maturity. That means they're no longer living when they're ready to work. And the reason that is advantageous for a plant is one, if they're no longer living, they can get rid of all their cytosol and their organelles. They're a hollow tube. Why is that advantageous for the movement of water? It goes faster, yeah, and it can adhere to the sides of these xylem vessels and have this really long column of water. Remember, water is cohesive to itself. Okay, so these cells are no longer living when they're actually functioning. Now, at some point they were formed, and when they're immature, they don't function as a water transport vessel at that particular time. So tracheas and vessel elements are the two types. Anyway, they're usually the vascular tissue for the xylem is usually found in the middle of like a tree trunk, the very middle. The phloem vessels are the ones that allow sugar stuff to move on the outside, and we'll talk about that in a minute. These function, like I said, in water transport, but they also can function in support to help a plant stand up straight. Okay, especially big trees. You think cell walls are important, but like a big tree, like a cell wall is not going to allow a whole tree to stand up. They have a lot of other stuff, including the bark of a tree and all that. There's a lot of lignin, and anyway, these help in support of a plant. They align end to end. Notice like when one cell ends, the other one begins. They have these little like plates, sort of like these discs that have holes in them that are perforated to allow water to move from one cell to the next. It doesn't become one long continuous. There's still different cells or like remnants of cells, so they still have sort of a beginning and an end. But they have perforated plates that connect them. Okay, now the phloem tissue is what the sugar solution travels in. And these are called sieve tube elements. These are actually living at functional maturity. But to maximize their space inside, they really sort of, all their organelles and everything they need is in a cell right next to them called a companion cell. It's their little best friend. Aww. So the companion cell produces all the stuff that they need to keep living, so they can basically just be a hollow tube that transports sugar stuff. So these are sieve tube elements, and they are also connected by what we call sieve tube plates from end to end that are also perforated to allow the sugar solution to pass through. There we go. So that's good.
Does McGraw-Hill have anything on this?
Probably not. It'll have transpiration stuff, but we're not there yet. Okay. Great. Take a deep breath. Let's keep going. This is a lot of stuff. All right, so plants. We're going to talk about transpiration first. The movement of water in the xylem is what we're going to talk about today. We'll probably do phloem tomorrow. So plants move huge volumes of water from roots to shoots. Mostly during the daytime when the sun's shining. Why don't they really need a lot of water moving around during the night? They can't photosynthesize. So why do they really need water if they're not really undergoing photosynthesis? They kind of do, honestly, yes. They do. Their little phytochromes turn off. They kind of do. Crazy. Okay, we've already talked about this. Most water and mineral absorption occurs near the root tips where the root hairs are located. Yes? The root hairs are most of the surface area. Increase the surface area of the root tips so they can get a lot of water and minerals in them. Yay! Okay, we've already talked about this. Good for us. We talked about the endodermis, this Casparian strip, the two roots. That's exciting. So the water comes in from the soil solution into the cell wall, and whether it's selected for or not at the beginning depends on what route it travels in. So it can flow through cell wall, and get in a strip where it eventually has to move into the cytosol, or it can go ahead and right away and get into the cytosol. Either way, eventually it'll be selected for. There's different types of plants, monocots and dicots. They have different anatomy. You no longer need to know that for the AP exam. They used to put diagrams like this, and you have to identify the type of plant. Thank goodness you don't have to anymore. Okay. So we've already talked about all this. All right, so let's move back to transpiration. You need to know that transpiration is a negative pressure-driven force. Water is pulled through a plant, sort of like a syringe, like you suck up water being pulled. What generates the pulling force is the evaporation from the stomata. That generates the pulling force on that entire long line of water molecules. The xylem sap is the liquid stuff that's moving through the xylem vessels. So is it mainly pushed or pulled? It's pulled. And like I said, that pulling force is generated in the leaves as the water is evaporating through the stomata. And we'll talk about why in just a second. Before we do that, let's think about a plant at night. And let's think about a really short plant. A little short plant. So at night, roots still work. Roots are still pumping out hydrogen ions, taking in minerals, all that. That happens at night. They don't know the difference between night or day, really. So, because they're underground. So it keeps happening. Well, what happens to the water? If they're still taking in minerals, what happens to the water? It's still coming in. It still comes in. So at night, in plants that are really short, enough water will come in to actually generate a very small pushing force from the root. And that pushing force can push water up just a really short distance. So in the morning, on some plants, you can actually see water droplets like this, hanging off the ends of leaves. This is different from dew. Dew is just, this is a little different. These little water droplets are actually force. It's generated by the roots working at night, bringing in the solute, the water comes in, it pushes the water up a very short distance. This is called root pressure, and then the exuding of the water droplets is called vegetation.
So like,
There's a question on the test on Monday about vegetation and how it happens.
yeah, so like a syringe, like before you make like when it will drop it into the needle.
Yeah, like a pipette here, I just dropped it. I can.
It's like you have a full syringe and you're pushing.
Yeah, that's what
Yeah.
And like, if you like, I don't know, it might just be a TV thing, but they get injected, like a little drop at the end of it.
Well, the reason they do that is because they want to make sure there's no air bubbles. So like I have to give myself shots, and whenever I pull the needles out, I have to like tap them to make sure the air bubble inside the medicine comes to the top, and I have to like squirt out the air bubble before I inject myself. So I always have a little like thing on the top of mine too, because I'm like making sure there's no more air. Yeah, I mean same kind of thing, pushing.
What happens like if you inject it with the air?
So if I inject it into a blood vessel with an air bubble, that's bad, because air bubbles and a liquid can cause something called cavitation. And like in a plant, like we'll talk about that, but you don't, water, like you have these long columns of water moving through your body just like you do in a plant thing. So in a really tiny blood vessel, it can actually cause an embolism. It's like a, yeah.
Oh wow.
It's kind of, the reason I have to do it in my legs because there's a lot of fat tissue, like on these people's legs. So you're much lower likely to inject it into directly into a vessel. Yeah, so you get out the air bubble. You can kill some mice. And yeah, I'm pretty sure. I won't look over my computer there. But you can look at injecting air bubbles into blood vessels.
Why are you making me?
I think that you have to kill them. Yeah. It's an embolism. It's called arterial air embolism. Same kind of thing. It blocks blood flow. It gives you a stroke. A heart attack. Let me kind of crazy. But all right. So on your board, you're gonna have to put a diagram and talk about what we call the transpiration cohesion tension mechanism. So let me talk about it now.
You said that in one breath? The transpiration cohesion tension mechanism.
How does this work? So it's a really long name for just how this happens in plants.
Yeah.
I'm going to talk at the beginning of the root.
Oh.
So in the root, here are our root hairs. They pump out hydrogen ions, bring in solutes. Water will flow in naturally via osmosis. Okay, water will come in and eventually move into the xylem vessels. Okay, now if we start here and think about water potential. Water potential is more negative inside these root hairs because there's more solutes. So it moves from a high to a lower water potential. And it becomes more negative inside the xylem vessel. And as it moves up a tree or a plant, notice what happens to the water potential as you move up. It becomes more and more and more negative, which is why it keeps moving up and up and up and up. Okay. So here we get into our xylem vessels. Now what happens in here, it forms a column of water. because the water molecules are hydrogen bonded to each other through cohesion, and they're hydrogen bonded to the walls of the xylem vessel through adhesion. So here's our cohesion, adhesion part of it, okay? So they form this really long line of water molecules. Now, if a plant is actively growing, now the wintertime, a lot of times plants go dormant. So they're kind of like hibernating, kind of. During the summertime, or the growing season in particular, why having a frost is bad for plants is that they're actively growing. When a frost happens or a freeze happens, what happens to water when it freezes? It expands, and then when it thaws, it contracts. Well, it can cause air pockets to form inside these xylem vessels. That happens, you broke that line of water, and now it's no longer going to come back together. So this entire column xylem vessel now, the whole thing from bottom to top is useless, and the plants can't use it anymore. So that's what could potentially kill some plants, is if you have cavitation of all the xylem vessels, it'll kill the plant.
So is that why they genetically modified the strawberries to prevent cavitation?
Well, sometimes it also damages the flowers. So the flowers themselves are really fragile, and if they frost and die, then a fruit will never form. Okay. Once we move our water column up to the leaf, so this is supposed to show a leaf, all right? So this is green now because these are mesophyll cells that are photosynthetic. The water molecules will leave the xylem and go into what we call the air spaces in between the spongy mesophyll cells in here. And they're still connected by cohesion. And then as they get close to stomata, they will evaporate. So when they do that, that pulling force will be transported like back to the entire line all the way down to the tree. So this is what we call the tension. So it's pulling. So now as you get enough kinetic energy, that actually breaks and go into the air. Oh. They will transpire faster. Yeah. Yeah. So we talked about that. This doesn't cost any energy to plant. Yeah.
So what happens when like you overwater? Or too much water is available?
You drown it. So the reason why you can drown a plant is because the roots, they aren't photosynthesizing. They actually rely on the shoot system for sugars, but the roots are undergoing cell respiration because they're constantly growing, so they need oxygen. If you overwater a plant, you get rid of all the air bubbles in the soil, and then does that make sense? Which is why plants, like we talked about, some plants that live in swampy areas, they actually have roots that come out of the ground or the swamp to get air. Okay. In the mesophyll spaces, in a leaf, as the water is evaporating, so all these mesophyll cells are surrounded with a thin film of water. That's this blue here. Okay. The higher the evaporative force, the more it's going to be pulling on these air molecules, or the water molecules on this film of water. And you can actually, your book has a picture. Where is it? This picture in here. So here's a film of water on these cells. So notice this film is pretty straight, not a really strong evaporative force. As evaporation gets like more quick, notice this becomes more and more like, words are not coming to me today. Curves. All right, the tension force is much higher here than it is down here. Anyway. What type of environment do you think would cause a plant to do this versus this? Where would plants transpire the most? Have the highest rate of transpiration? When it's hot? Humid? When it's dry. Arid, hot environments will cause water molecules to evaporate much more quickly. Okay, so think about, so that's sort of the basics of your lab. So the people that are doing the different like temperatures, so these little plants over here under this heat lamp, it's causing the water molecules to have a much higher kinetic energy faster, right? And so they're going to tend to leave faster than the ones that are in like an ice bath or the ones that are room temperature. And was it your class that's doing humidity? Or was it the other class? Somebody in the other class is doing humidity over here with the bags on their plants. So the more humid an environment, water is not going to tend to evaporate. Have you ever? Okay, so we live in South Carolina. You know what it's like in July and August? You go outside, your clothes like stick to you. Why are they sticking to you? And why do you have like sweat rolling down you? Oh, it's humid. And the water's not going to evaporate off of you. So it'll just like beat up on you and like come out because you're still going to sweat. You go out west, however, it can be 110 degrees and you're like, it feels like it's 85. It's great. It evaporates so quickly off your skin, it cools you down much more efficiently, and you don't feel it.
Yeah. You know that group's back there? Did theirs die?
Yeah.
The one with that big
The one
heat
that's
lamp?
hanging?
I mean, it's not a heat lamp, but like it was.
Yeah, it was so close to it, it melted the bottom of the plastic.
Oh, yeah.
So we discontinued that.
They have to start something new.
They still have 2 other levels, so they're going to continue with their other one.
Oh, I don't know why you didn't try that. I could.
They can't. They lost soil, so they already lost mass that wasn't just water. So they're not going to be able to. Okay. Yeah. Go ahead.
If someone's doing an experiment on the water, they have on quicker than they don't have the whole water, access to the whole water.
Right. Yeah. I mean, some of y'all labs are going to feel the same. Like the wind groups are probably going to die. Because what happens to wind? Wind will also, it'll dehydrate because the water will evaporate more quickly.
That was the fan.
Yeah. Yeah. Okay, let's see if I think I have a video I can show you now. It is.
Yeah, you did. Yes, you did.
Oh, they don't have it.
Yes, I'm so
Water and nutrients need to move throughout the entire tree, even against gravity. How does a plant manage this without an organ like a heart that pumps fluid? As we will see, water is poured from the roots to the leaves through a process called transpiration. In addition, water potential drives the movement of water from one area of the plant to another using osmosis, gravity, and the surface tension of water. Transpiration begins in the leaves. The arrangement of cells and structures in the leaf facilitate the movement of gases and water into and out of the leaf. A leaf contains several layers of specialized cells. The upper epidermis is one cell layer thick and provides a protective covering. Below that layer is mesophyll tissue. Cells in the palisade mesophyll layer are sites for photosynthesis and secretion, as well as storage of food and water. The spongy mesophyll layer contains a looser arrangement of cells, where spaces between cells aid in gas exchange and the passage of water vapor from the leaves. Throughout the lower epidermal tissue are stomata, which are microscopic openings flanked by guard cells. Gases pass into and out of the leaf through these openings, as well as water vapor evaporating from the leaf, a process known as transpiration. The spongy mesophyll layer contains arrangements of vascular tissue, consisting of xylem and phloem that are specialized for the transport of water and nutrients throughout the plant. The vascular tissue extends from the leaves, through the stem, to the roots. Water is transported in xylem from the roots, where the water potential is higher, up to the leaves, where the water potential is lower. The arrangement of the tissues, the functions of the cells, and water potential determine the direction in which water will move through a plant. Water passes out of the leaf as water vapor through the stomata. The water vapor lost from the leaves is replaced with water that enters through the roots and is brought up through the stem in xylem. Xylem is composed of vessels, which are continuous tubes formed from dead, hollow, cylindrical cells arranged end to end, and tracheids, which are dead cells that taper as the ends overlap. This arrangement and the polar nature of water molecules allow water to pass in an unbroken stream through the xylem, from the roots up through the shoot and into the leaves. Adhesion is the attraction of water molecules to a surface, such as the wall of the xylem. Cohesion is hydrogen bonding between water molecules. Together, adhesion and cohesion allow water to move through the xylem in a continuous stream, from the roots up through the stem to replace water lost from the leaves through the stomata. Water enters the plant through the epidermal cells of the roots and travels into the xylem. Water potential in the cells of the roots increases when symporter pumps in the plasma membrane allow protons to pass into the cell, traveling down their concentration gradients. These pumps couple the transport of protons with the transport of minerals and other solutes into the cell. Water follows into the cell, driven by osmosis. The presence of aquaporin channels in the membrane enhances osmosis, allowing bulk flow of water from the soil into the roots. The other main vascular tissue is phloem. Phloem transports carbohydrates and amino acids that are produced in the leaves to cells in the roots and stems where they are used and stored. Conduction in phloem is carried out through two kinds of elongated cells, sieve cells and sieve tube members. Most angiosperms contain sieve tube members. Both types of cells have clusters of pores known as sieve areas that are abundant on the overlapping ends of cells. These structures aid in the movement of carbohydrates, like sucrose, that are manufactured in the leaves and carried in the phloem throughout the plant, a process called translocation. Turgor pressure increases in the sieve tube members as sucrose from surrounding cells is brought into phloem through active transport. Water then enters phloem from xylem by osmosis, which drives the transport of carbohydrates in the phloem. Water movement in vessels is one way, while transport in sieve tube members can go in both directions. Water potential is an important driver in both xylem and phloem transport, but only phloem transport utilizes both active and passive transport. Our heart pumps blood throughout our bodies to provide nutrients and water to our cells. Vascular plants can accomplish this same feat.
Huh? Oh, where were we? Okay. So to make sure we know, be loud. Eloise, I'll be back by 6 if you all want to do stuff over here. That was my ADT. I'm going to come like after we write in class in the 2. Okay. We talked about this already. We'll go with how water comes into the roots and then how minerals comes in. We're good? Okay. Great. All right. Talked about the mycorrhizae. And let's talk about the stomata and the regulation. So the guard cells are the cells that will regulate the opening and closing of the stomata. So how does stomata actually open and close? Well, the structure of the stomata, or the structure of the guard cells, is super important. First of all, let's look at this. You see how these cells have these little lines going? These are cellulose fibers, and they are wrapped in a particular orientation around the guard cells, such that when the guard cells become turgid or full of water, they will expand, but not in every direction. They will expand in such a way that causes them to bow, like a lima bean. Okay, so they open. They create an opening in between. And when they lose water, they will become flaccid and they will close the opening. See that? Yeah? All right, so what causes them to become turgid or flaccid? It has to do with the movement of potassium ions, which are the red dots in this picture. So when a cell has lots of water, they are transpiring, but they have what they need, and the cells are turgid. Potassium ions will move into the guard cells, and then water will follow via osmosis, and they will become turgid. So when a plant is losing too much water, or it's at nighttime, for example, a signal will be sent to cause the potassium ions to leave the guard cells, and then water will leave via osmosis. Then they will become flaccid, and then they will close the stomatal opening. See that? Okay. So potassium movement will regulate water movement, which regulates the tonicity, or how turgid they are, and then will open or close the stomata. So how are the potassium ions taken up by the guard cells? It has to do with proton pumps. So the proton pumps will pump out, and then the potassium ions will flow in, and then water will come in through osmosis. Okay. Yay. And then when potassium ions need to leave, they will just flow down their gradient through channels, and then water will leave. Yay. So stimuli for opening and closing stomata have to do with time of day and other hormonal control. So first of all, stomata mostly close at night because there's no light, there's no photons, there's no need for extra water, there's no need for photosynthesis. So there are proteins, the phytochromes, that will change their shape when they get a certain wavelength of light. So when the sun starts to rise, some of those phytochromes will change shape. When they do that, it will indicate like, oh, the sun's rising, and so the plant knows, all right, it's time to send potassium ions in, we need to open our stomata. It can also be triggered by carbon dioxide depletion. So when these photons are starting to be absorbed by the plant, they will start undergoing photosynthesis. When they do that, they'll start to use up the CO2 that's in the areas around the mesophyll cells. So when the CO2 levels start to drop, that can also signal the guard cells, hey, it's time to open. And also, there's something called circadian rhythms that all eukaryotes have. It's an internal 24-hour clock. It kind of tells all organisms what general time of day it is. We have it, plants have it, every eukaryote has it. It's kind of crazy. We'll talk more about circadian rhythms later on. So, it's important to balance stomata opening and closing with water loss, but also with the need for carbon dioxide and the need to get rid of excess oxygen. So our plants can undergo photorespiration if there's too much oxygen and not enough CO2. But they also don't want to lose so much water through transpiration that they're going to start to dehydrate. So there's an important balance that plants have to have. Plants lose a huge amount of water through transpiration. Topsoil, get it? Topsoil.
What's that one over there?
If you haven't been taking your vitamins, what have you been doing with them? Like she's talking to the little boy and looking at his giant plant over there.
Oh.
Okay, what's the next one?
Cartoons, like plant cartoons are not funny.
About 20 years ago.
Fletcher, so when he was little, his mom would give him like a vitamin, and I guess he hated them. And he would take him to his room, or I'd put him in his mouth and pretend to eat him, and then he'd take him out and put him under his bed. And when they moved houses, when he was like 7, she like said they moved his bed, and he had this giant 5 billion cluster of like hardened like Flintstone vitamins under the like in the carpet under his bed. And she was like, "Oh my gosh."
No bugs, no indication that
I don't know.
there's.
I don't know. Yeah, it's kind of. My kids love their vitamins. They love them.
I mean, they make them taste better now.
Yeah. Right.
But I remember I had like everything just like things like stuff like that. So I had like a container. We went to like 5th grade and we went to the candy shop that had something like that. And I accidentally left my bag because when I got home I just threw everything in my closet. I accidentally left the candy in there. Like a week later we had like ants, like little
Oh
ants.
yeah.
So I was like, what is it?
Oh,
She
great.
was so bad at me.
Okay. So plants, we don't want plants to wilt. What's interesting is plants can use evaporative cooling through transpiration to help cool themselves down, interestingly enough. So Remember, some plants have adaptations to prevent transpirational water loss by living in really arid or hot environments. Xerophytes are plants that are adapted to really dry or arid climates. So a lot of these are cacti. They have leaf modifications to reduce transpirational rates. So some of them use crassulation acid metabolism. We talked about that last semester where they only open their stomata at night. Remember that? Okay. So some of them, this little plant only produces leaves right after like a rain. And then it has little tiny, they're so super tiny. Look how tiny they are. Leaves, and then they'll flower really fast and they'll get rid of them. So it helps prevent water loss, but they only will produce them after a sort of significant amount of rain. So
What are the little things?
an oleander plant has so under their leaves they have these things called crypts, and it's like an indention of its leaf. And in the crypts are where the stomata are, and they even have little hairs called trichomes. The trichomes sort of function as like the hairs on a cat or a dog, or when it stands up, to kind of increase humidity in the area in there to help reduce water loss. It's kind of crazy. Anyway. There's a cat. That thing was hard. You touched it though.
It looks like.
What are those made
I know, it
of?
looks tough.
What are the little
Those are all the
pine
little
things?
like spikes. Those are all the like the
Yeah, I know. What are those?
they're modified leaves. It does look like a cattail.
I like that.
Okay, I think I'll talk about phloem tomorrow.
I'll put vodka in there, though.
What?